check on the website, not an issue. On 13th, we have Dr. Neeti.
I'm going to start the broadcast, okay?
Okay. At that time, check on the website.
So what are you? তুমি তো mute করতে পারবে? So what are your plans today on your birthday?
Not much at home.
Yeah.
We'll cut an ice cream cake. It's very hot. We are not cutting a usual cake. We're cutting an ice cream cake. Not much, nothing. Calls, video calls. Nowadays, my life is more
Yes.
into video calls
Yes.
with everyone.
Yes.
Because people are more interested to see the hand rather than me. So,
yeah, he will be
usual, yeah.
getting all the limelight. Yeah, star of the show and everything. Little Trishan.
Yes.
And from now on,
yes,
I think all your birthday cakes will
so
be
we
cut
have
by
got
him only.
Yes. I think you're telling this from your experience.
Yes. Cut, smooshed, mashed.
Ori Baba. You are scaring me.
All right.
How?
So very happy for you. So you can go back and,
Thank you.
Australia.
Another, yeah, but I'm a bit quite scared also. Leaving the
and all,
comfort
yeah.
zone and going on your own.
Yeah.
Traveling and everything. Let's see
A whole
how it
new
goes.
story now, right, with Trishan?
Again, a new whole story. Again, it will be like starting from scratch. He's so used to people around here and no one will be there.
Yes.
So
even yeah.
So it will be a problem.
We're in live.
Okay. So it is really hot here. It's really hot. Very, very, very warm and hot here.
Okay, I'm back.
Hi.
Hello. So how do you do it? Like from Zoom, you go to YouTube?
Yes.
I don't know. I'm not technologically that great.
তুমি unmute করো না।
Actually, we broadcast. There is an option to broadcast the event. So we broadcast it on YouTube, and when we suggest all our participants to follow us in YouTube, and also I send all the participants a mail that this is the live link. And before that, I also give them intimation that we are going to live.
So when do you start? When you see?
So you are still collaborating with Dr. Neeti?
Nidhi at present, no, because now we are doing completely different things. In future might be, because now we are working on synthetic biology. So more like high value products, chemical products, and algal biofuel.
Okay. So there is, yeah. So you would be feeling some change between the industrial way of working in life sciences and the academic side where you were earlier.
Previously, I was with Syngenta.
You have more rights and
yeah,
I'm aware of that.
So where you guys work now?
No, right now, I have to go back Sydney. I came India.
Hmm.
So I did my PhD with Dr. Neeti. Abhishek, who was your postdoc student, was my junior in the lab.
Okay.
Abhishek Anand.
Why you are calling Nidhi?
Yeah.
It's not Niti.
So through that, Neeti.
Okay. Yeah. Yeah.
I'm
So
saying Neeti.
yeah, Abhishek is
Yeah.
now.
Yeah.
Last when I talked to him, he was in
In
Taiwan, and then probably he moved to somewhere else, or I'm not sure now.
Yes. He's still there, locked up due to COVID-19. He's trying to get out and go to US. I think he has got his visa. So soon he will be traveling. He was all stuck because of the travel restrictions going on.
Yeah, he asked me for a recommendation.
I
I
just
sent,
had a
but
chat with
later
him
on I
recently.
didn't hear from him. So I was not sure what exactly
Okay.
happened.
Mhm.
Roma and Subhabrata, you are
Good.
where
Yes,
are you?
I'm working in Calcutta University, Dr. Shapan Kumar Datta's lab.
Mm-hmm.
And now my lab is under lockdown. We are trying to open as soon as possible. Just sanitization is possible, but university will open in, I think, in October.
Okay.
So that is also a good time to start Bioingene. I got time to arrange.
No, it's a very good initiative, and most importantly, so much of response.
Yes.
So that's good.
Yes. Okay.
I think this platform has, deleted the need to travel all through for one seminar. So people
Yeah.
can just sit and listen to you big shot people.
Not
Thanks
big
to you
shot.
guys. And then,
No,
so,
problem
the
is
ease of technology has actually helped
lot of
get.
webinars and seminars are happening, but those are all paid. You have
Yes.
to, and you
Yeah.
guys have kept it
Yeah.
free and etc. That attracts students. That's good actually.
Do you need
Yeah,
the
and
YouTube
through
link?
this we have.
Pardon?
Do you need the YouTube link? Should I send you in LinkedIn or it's okay?
No, what I would
Okay,
do with
no
that?
problem. No
I'll
problem.
present here and rest part is yours.
Okay, no. I think if you need the link or all. Okay, we are set now. We have sent all the in. We are now live also in YouTube. We got the responses. There is a lot of people now watching us. Is 202 people are watching. So should I start? Okay, 18. Welcome everyone. Today is our 18th webinar on this series. I welcome all of you to our Bioingene platform. Today we have our speaker Dr. Anindya Bandyopadhyay, and we have Dr. Neha and Shoma. I request Neha to introduce our speaker.
Thank you, Shiva. Welcome to the Bioingene webinar series. Today is our 18th webinar of the series. Bioingene is a non-profit online organization created to promote plant research worldwide. We are very happy to organize plant research webinars that are free for the whole world. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope you will gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant science. We are grateful that many of you have joined us today. For today's webinar, we have received a huge response of around 4,000 registrations. We welcome all the viewers and thank them for their interest in Bioingene. After the talk, you can apply for a certificate of participation via the feedback link and password which will be provided after the question and answer round. Submission of the feedback form is mandatory to receive a certificate. The feedback link will be provided in the YouTube chat box by the host. Please be patient. You will get ample time to fill and submit your feedback form. Please collect your participation certificates after 2 days from our website. Please visit our website to register for upcoming webinars. You can connect with Bioingene via Facebook, YouTube, Twitter, and LinkedIn. So the topic for today's webinar is CRISPR in agriculture, context of improving water use efficiency by Dr. Anindya Bandyopadhyay, Vice President, Reliance Industry Limited, India. So Dr. Anindya Bandyopadhyay is a genome editing specialist and presently leading the synthetic biology and genome editing program in Reliance Industries Limited. Previously, he worked as the Global Genome Editing Program Lead for Syngenta. He was also faculty and group leader of the molecular biology in International Rice Research Institute, Philippines. Anindya did his PhD from IRI, followed by postdoctoral training in the University of California, Berkeley, and University of Kentucky in the field of plant molecular biology, genome editing, and virology. Research papers, reviews, and book chapters. He is also serving as a reviewer in many renowned international journals. We are very happy to have him with us today. Sir, kindly share your screen and proceed with your talk.
Hi, am I audible?
Yes.
Okay, great. And you can see my slide, right?
Yes.
So, good morning all, and thank you Bioingene for inviting me to give this talk. So I realized almost 4,000 people have registered, which is amazing, and mostly a big number from students and postdocs. So after talking to the organizers, I decided to change my talk a little bit, where 50% I talk about the basic CRISPR and how this could be applied in different fields. And then I switched to agriculture and eventually a case study with water use efficiency. So if anytime I am inaudible or link goes, please let me know. I will try to do something. I don't know what I will be able to do, but I will try. So thank you again, Shubhabrata, Neha, Shoma, and especially Neha for introducing me here today. So, oh, it's not moving. Yes. Just a second, let me find a good pointer. Laser pointer. Okay, so I have divided my presentation in, I would say, 6 areas, rather 2 broader areas. So when first I start with the history of CRISPR development, then some basic applications, basic science behind CRISPR. Then I talk about application of CRISPR in different fields, and then recent discoveries and what kind of modifications or advancement is going on in the field. And then I switch to agriculture, how this plays an important role in agriculture. And then finally give us case study on water use efficiency, how CRISPR could be used in plant to develop drought-tolerant plants. So before going to my presentation, so I'm coming from Reliance Industries, and Reliance Industries is one of the biggest conglomerate in India, and with international presence, and one of the Fortune 500 companies, the biggest conglomerate here. And mostly 3 areas Reliance Industry works. One is geo data platform, 4G mobile internet telecommunication. This is a big area of Reliance. The second part is Reliance Retail, where diversified product portfolios, groceries, fashion, lifestyle, etc., consumer electronics, connectivity, etc., are involved. And the third, one of the major area of Reliance is providing transport fuels. So basically aviation fuel. So and supplying petrochemicals to diversified end-use industries, manufacturing essential industrial and consumer goods using polymers and polyesters. So we come under a R&D, which is within the jurisdiction of petroleum business, the third one. And our major interest is to develop biofuel. So we are working on algal biofuel last 6 years with state-of-art biggest biofuel, algal biofuel plant, which is shown here. It is in Gujarat, and our laboratory is in Reliance Corporate Park, Mumbai. So, and this is basically algal plant where you see all different types of ponds, greenhouses, and different computational manipulation of this whole thing, and everything is computerized here. And we develop algal biofuel here and do different kind of research. And also we are using algae. After taking out the oil, we are using the remnants for different high-value chemical production. And as you understand, CRISPR is nothing but applying a new tool, RNA and protein-mediated tool for cellular modifications. So we try to modify algae with CRISPR for different purposes. And also we are targeting some of our activities in the select agrobiotech research. So this is where I'm coming from. And now let's start with my first area, CRISPR craze, how CRISPR started and the basics and the applications of CRISPR. So far, whatever we had was random transformation. That means we can send a gene of interest or a molecular tool inside of a chromosome, which is having DNA, double-stranded DNA, by random transformation techniques. So it can go anywhere. You cannot control. Either you do transfection in animal or human cell line, or you do plant transformation by agrobacterium or bialistic. You send it randomly inside of the genome, and then you select for a good event where you get a desired trait. But it's random, you cannot control it. So since the advent of this CRISPR-Cas9 discovery, this technology and discovery of CRISPR-Cas9, we have now capability of targeting a particular region of a genome, and we can change that genome selectively as per our will. So this is a big discovery in biology, and also I would say one of the greatest discoveries in last 1 decade for overall technology field. So MIT Technology Review has considered it one of the most important technological advancement in overall technological aspects. So we can do targeted change now. We can target a region, send our genome editing instruments, and can edit a particular region. So once this CRISPR was discovered, almost 6-7 years back or 8 years back, we started seeing a boom in different areas, like funding shoot up in 2013 or 14 area like anything. Private sector as well as government, they are putting a lot of money so that this field can progress faster. Also, if you see different kind of publications, then investment by the venture capitals, and also the patents, all are shooting up rapidly and very significantly in last few years encircling this CRISPR-Cas9 field. So CRISPR-Cas9 first discovered by two competing institutes. One is MIT and second one is Berkeley. So I tried to show this. So MIT came up with CRISPR-Cas9 application in eukaryote, and Jennifer Doudna's lab in Berkeley, they came up with CRISPR-Cas9 can do editing in prokaryote, the primitive cell types. And in first editing efficiency of CRISPR was shown by Jennifer Doudna's lab in Berkeley. And these two labs, then what happened, you could see many big companies started taking license from them. So Dow, Novartis, Horizon, Yet Genomics, DuPont, Intelia, Bayer, Horizon, GE, Taconic, many Sigma Ulrich, many bigger companies started sub-licensing it, and a big craze of using CRISPR for myriad applications started coming up. So in global CRISPR-Cas9 gene market to surpass 7 billion by 2026. This is the recent Bloomberg prediction. So it's quite a craze. Now, what is CRISPR? So, and how it was found? So if we see this whole thing, I would concentrate here, a yogurt plate. So if anybody, whoever has eaten yogurt from shopping mall, there are many markets you can go, you can find small packet of yogurt, which is called dahi in India or curd. So whoever has eaten that from a shopping mall, they have eaten lot of CRISPRs already. So we are eating CRISPR since last 20 years. So what is that? Basically, dahi or yogurt and all this cheese, etc., they are made by application of certain kind of bacteria, right? So these bacteria are used in this dairy industry since long back. And they found one interesting thing, that some of the bacteria can resist fudge viruses. So bacteria has natural enemy, which are basically fudge viruses. So they can resist those viruses. So scientists started, R&D team of all these yogurt companies started culturing those bacteria only, which has these intrinsic capability of resisting the fudge viruses. And later on, after doing the molecular analysis of those specialized bacteria, they realized these bacteria are having certain kind of strange sequence inside of their genome. So this whole cell is a bacterial cell, and you can see a fudge virus is attacking a bacteria. Once fudge virus attacks a bacteria, it inserts its genome inside of the bacteria. So bacteria get sick due to the attack of fudge virus. But one thing it does, it cuts the viral genome and internalize that small fragments of the genome inside of its own genome and keep it as a memory. So next time when that same virus or same group of virus attacks this same bacteria, it already has that memory of attack, and then they make certain kind of tools, that tools basically designed from this memory and destroy the attacking viruses. So this is nothing but memory dependent bacterial adaptive immune system. So these memories, these tools which I'm talking about, this memory dependent tool, these are nothing but later on people realize these are nothing but CRISPR-Cas9. So these CRISPR-Cas9s, then scientists used to find out that this can be modified in certain ways, so it can do targeted gene integration, targeted gene editing. So the story I was talking about, Danisco was one of the companies of dairy companies, which basically used to culture all these bacteria. Later on they found, and many other companies also. So later on Danisco was acquired by DuPont Pioneer, and then and Danisco's famous strain, Choose It Swift. This is basically a bacterial strain. They sell to different companies, yogurt companies and cheese companies. They use this Choose It has nothing but CRISPR arrays inside of their genome, and by this CRISPR array they can get rid of viral infection, so that they stay healthy and they make good yogurt and good cheese. So I took some time to describe this interesting aspect of discovery. And later on, it was found that there are different types of CRISPRs that are available in the system, in the nature. So mostly people divided into two classes. One class use one RNA and a big protein called Cas protein. It can target DNA, it can target RNA. And there is another one class known as class 1, which has many different proteins, different cascade of Cas gene and RNA. So these RNAs are called guide RNA. So two types. This class 2 type is majorly used for genome editing purposes. So bottom line is, what is CRISPR? It's nothing but a gRNA, these lines, and this green one, a protein, which is called Cas9. So only two things, gRNA and Cas9 or Cas protein, they get fused together and they can be used for genome editing. How that happens? Again, in this left-hand side picture, I show that this is the Cas9 protein, and this is the gRNA, this filament. And then they go and sit in a double-stranded DNA. And what happens? So first, this gRNA and Cas9 come together, they make a complex. That complex goes and sit in the double-stranded DNA of any organism, any living organism, or which has a DNA inside. They can go and sit there, and then they cut it. They give a double-strand break. Now, cell cannot keep its DNA or its genome as a break one, or a one which is already got certain kind of break, double-stranded or single-stranded or whatever. Now, cell wants to repair it by its own repair machinery. And while they try to repair it, they make some mistakes. They bring some mutations. And that mutation gives rise an editing. What is that editing? That mutation can switch off a gene to work. How this could be used? If we have a gene which is giving rise to a certain kind of protein which is not good for our health or not beneficial or harmful, you can use this CRISPR-Cas9 system to target that gene and cut that gene in a double-strand break, and then cell will try to repair and will bring some mutation, then that gene will not work. So any hereditary disease or any disease which is caused by a certain gene, we can basically knock that gene off. We can basically stop that gene from working. So this is one way. And the other way is, similarly, you take Cas9 and gRNA, make the CRISPR-Cas9 tool, let it get inside of the genome and cut the genome, and then you supply a rectified gene there. Then what happens in the cut region, that rectified gene gets in. So you can insert a new gene from outside, inside of the genome, in a more targeted way by using this tool. So these going in process is known as homology-directed repair, HDR. What is the beauty of this system? It's very easy to use, very cheap. Only two components are needed. You can design this gRNA component, and based on that, you're designing that gRNA, go and sit somewhere and do your work. Now, you can use many gRNAs. So in one shot, you can target many regions of the DNA, many regions of the genome, and you can do multiplexing. So in one shot, you can do change in many parts of the genome, or you can change many genes. So this is what I showed here, that CRISPR-Cas9 goes, sit there. Two ways it can do the change. One, it cuts and cell try to repair and bring some mutation, and that gene stop working. So gene knockout is possible. And you cut and put a stretch of foreign DNA or foreign gene or gene from any other organism, or a gene from the same organism within the same genome, and you can insert that by homology-directed repair by using this CRISPR-Cas9 system. So this was the basic. People identified a bacterial adaptive immune system, which basically uses a thing called CRISPR-Cas9, a tool that can be used for genome editing, mostly change the gene activity or switch off the gene or insert a new gene there. But later on, with protein engineering and different kind of molecular biology activity, people started to modify this system and they could do many things, such as epigenetic regulation. As we know, apart from the gene and genome, there are other regulatory things which are basically known as epigenome. They can do methylation or certain kind of changes, or induce histone acetylation or demethylation, whatever, and they can do targeted epigenetic change. So our genome, irrespective of the change in DNA, they can work differently. So one gene can work differently due to these epigenetic factors as well. So by a CRISPR-Cas9 system, with a Cas9, you can hook different kind of epigenetic factors. I mentioned here P300, LSD1, MQ1, TET1, etc. And you can even change the epigenetic pattern of a genome. Side by side, you are taking the Cas9 protein and you are taking the gRNA, only two components you are taking. Now in the Cas9 protein, you attach one activator molecule. So you can activate a gene. Or you attach one repressor molecule, you can repress a gene. So activation, repression, all can happen using this CRISPR-Cas9. Scientists came out with another new area, which is visualization, tagging, etc. by CRISPR-Cas9. It is more precise, more targeted. So the gRNA, I am always mentioning that CRISPR-Cas9, there are two basic things. One is one protein, which is known as Cas, and another one is a gRNA. So you put certain kind of aptamer or attach certain kind of aptamer in the gRNA, and then you hook a fluorescent molecule there. So what happens when you send that whole tool inside of the cell, you can visualize a particular area of your genome. So visualization, purification, when instead of fluorescent molecule, if you put a tag like flag tag or biotin tag or PA tag, whatever, you can basically pull that down and you do purification. And also by CRISPR-Cas9, there is a certain kind of CRISPR-Cas9, CRISPR-13 group of Cas9, CRISPR-13 group of system, which can even target RNA, which can make change in RNA. So DNA, RNA, both can be targeted by CRISPR-Cas9 and can be modified or adjusted as per your wish. Which exact area you wanted to target, you can do that by designing this gRNA. So if we see the modifications, so different kind of molecular modification could be done. So I already mentioned you can do knockout, both the strand break, double strand break. You can do a gene repression by attaching a repressor molecule. You can do activation. You can do tagging. You can do purify. Or you just attach a fluorescent molecule with your Cas9 and you can visualize an area. Or you can do genome-wide screening. And this Cas9 protein has two lobes of cutting. You can just delete one lobe, function of one lobe, then it will do a nicking. It will just cut a single strand instead of double strand. So those kind of Cas9s are called nickase. So you can do single strand cut, double strand cut, and many other modifications. So one can see a single simple tool of CRISPR-Cas9 can do many things, many changes, activation, repression, imaging of genome. And so one of the silver bullets scientists are considering and widely applying in different fields for different kind of benefits using with this CRISPR-Cas9 system. So in the next step, I'll go to these applications. So how this CRISPR-Cas9 is delivered inside of the research organism. So either a plant, human, or we work greatly on algae. So I have kept also algae here. So we use algae for biofuel and chemical production. So how this CRISPR-Cas9 tool, it's a simple tool, a protein and gRNA could be delivered inside of this human cell or plant cell or algal cell, whatever. So in human, there are two ways people are doing it. One is in vivo targeting, one is ex vivo targeting. In vivo targeting, they're using viral vector, liposome, polymers, peptides, and injecting, or even they can inject inside of the embryo and do certain kind of changes. And ex vivo targeting, what they're doing, they're changing cell lines in the Petri plates by CRISPR-Cas9 and delivering it by viral delivery or electroporation, whatever. And once the cell is changed, this is cultured in a greater extent and is injected in the bone marrow. So bone marrow transplantation, if certain kind of diseases happens in the bone marrow, you can replace that by transferring CRISPR-edited rectified cells inside of the bone marrow and save that person from a disease. So this is these are the ways it is used in human. In plant, there are two types, agrobacterium or bioelastic mediated transformation. Same agrobacterial or bioelastic transformations are taken, and CRISPR-Cas9 tool can be introduced inside of the cell. For algae, electroporation is done to send the CRISPR-Cas9 tool inside of the algal cell and do the change. So there are new areas are also coming out. We know about nanomaterials. So these nanomaterials are very minute and finite particles which can get inside of the cell very easily. Even by passive transfer, you can transfer nanomaterials inside of the cell, and you can attach CRISPR-Cas9 tool on the nanomaterial, and you can send that nanomaterial inside of the plant cell or animal cell. A lot of research is going on in that area. So we have CRISPR, now we know how to deliver that CRISPR inside of the cell. So once we deliver that, we can do many things. We can do many kind of, we can apply for many purposes. So for therapeutics, it can be used, gene and cell therapies, antimicrobials, animal health. We can do basic biology research. We can do agriculture biotechnology by doing precision breeding. We can bring novel trait. We can influence crop genetics by these, and also industrial biotechnology like microbial fermentation, therapeutic bioproduction, etc. I will briefly mention some of the big applications which are already happening. So this slide shows you how CRISPR application happens in human and for disease discovery, disease research, or gene therapy. So you can see cancer therapy, different cancer models could be made, animal models could be made by CRISPR-Cas9, cancer immunotherapy, genome-edited immuno T-cell can be made. And in human trial, already one such system in human trial in China, esophageal cancer PD-1 gene modified to resist cancer. So in fertility-related research, in vitro gametogenesis, gene knockout to stop miscarriage. In Francis Crick Institute, UK, people are using CRISPR-Cas9 to stop miscarriage of embryonic cells. Different genetic disease such as cystic fibrosis, genetic blindness, Huntington's disease, beta thalassemia, etc. People are targeting and trying to modify or do therapy by CRISPR-Cas9. Similarly, infectious disease can also be targeted in similar way. So University of Pittsburgh is using CRISPR-Cas9 to shut down HIV replication. A lot of work happening in Kobe University as well. And also organ transplantation. We know that different organs, animal organs are people are trying to use for human, like liver or whatever. So these organ development could be expedited or modified as per our need by using CRISPR-Cas9. Also in cancer, different kind of cancer models. I'm not going in detail. This will be available later on. One can see it. Different areas of cancer, this CRISPR-Cas9 can be used. And also drug discovery, CRISPR-Cas9 plays a big role, starting from target identification, compound screening, hit validation, lead identification, and finally clinical trials. So any compound you found, you develop animal model by CRISPR to test those compounds, or even it's a natural compound, then you can change the production process of that compound by using CRISPR-Cas9. So a lot of work is happening in this area, and almost more than 1,000 Cas9-based such gene drug discovery activities are under pipeline for the regulators. So it's happening very rapidly. Also, diagnosis, you know, this is our paper in preprint, which will come out in a quite renowned journal very soon, where we basically accumulated different kind of CRISPR-Cas9 based targeting of the COVID-19 disease, SARS-CoV-2. So CRISPR-Cas9 can be used by much faster in a point of care way for identification of different nucleic acid detection based disease detection. So COVID is one of them. Similarly, CRISPR-Cas9 people are using for animal model system and disease research. So these monkeys, rhesus and cyanomegalous monkeys, resembles humans in evolution, anatomy, physiology, and pathology. So they are widely used. So this picture is from one of the research outcome where BAML1 gene is knocked out. So circadian rhythm, our timing sense, is destroyed in these monkeys. So sometimes they sleep when in the daytime, or they are awake in the nighttime. So their circadian rhythm is changed, and people are doing many kind of research to find out how that works and what kind of drug could basically help in insomnia and other kind of diseases. Similarly, muscle-related things, these Beagle cloned and done by CRISPR-Cas9 for extra muscle growth, and different research is going on by using CRISPR for animal model system. Similarly, for pet breeding, like Dalmatians are very well-known and popular breed. They have certain kind of genetic mutation that makes them prone to suffer from bladder stone. By CRISPR, people have shown that these bladder stone related gene could be shut down, and similarly, faster horse breed can be made, healthier fish can be made. And in dairy, we know there are all these cows, they're having these horns and they fight with each other. So all dairy system farmers, they cut those horns, and very crude and brute way of handling them. But by changing a single gene, you can do the dehorning. And there are a lot of examples. People have made dehorned cows, so though they don't need to dehorn physically. And so it can be used greatly in animal and livestock. Similarly, what we are working at Reliance, we basically work in the industrial biotech area and select crop biotech area. So there are a lot of possibilities are there, starting from yeast, algae, in vitro enzyme production, plant cell mediated chemical production, filamentous fungi mediated chemical production. So all these bioproduction system in industrial fermentation, basically what happens, cells manufacture a product. So now detergent enzymes, ethanol, pharmaceutical drugs, vaccine can be produced inside of the cell. And CRISPR system can be used to make production cells better factories. So this is another area, industrial biotechnology, a big area we are working. CRISPR can be used in this area for terpenoids, alcohol, amino acid, antibiotics, lipids, anti-tumor agents, polyhydroxyalkanoids, phytochemicals, organic acids, many such thing development. We can use CRISPR from different kind of cell and cellular organisms. So CRISPR-I and CRISPR is greatly used nowadays in bacterial metabolic engineering as well for certain kind of product development. And as I mentioned, algae is greatly used. So we use algae, this is our farm, and we use CRISPR and other gene editing and other tool to change microalgae so that we can make bulk chemicals, biofuel, lipids, biogas, or pigments. So for different chemicals from algae and obviously biofuel by applying CRISPR. So far what I have shown is basically CRISPR-mediated different thing development, different important chemical development, and CRISPR. What is CRISPR? Its basics, from where it was discovered, what kind of modifications, what kind of application it could be done. And recently, there are many modifications have been made in CRISPR, which has wide application in animal, human, industrial biotech, as well as crop biotechnology or agriculture. So I will run through some of these modifications. So we know that if you take a Cas9 and hook one cytidine deaminase in the Cas9 and you send that target, what happens? It can change a single base. So single nucleotide polymorphism can be changed by attaching one deaminase molecule with the Cas9, which is nothing but we call as base editing. So base editing, there are 3 types of base editing can be done nowadays: C to T conversion, A to G conversion, or A to I conversion in a RNA. So different kind of further modification has been done to make it more precise. I'm not going in detail. All the references and some of the major things are here. Later on, you guys can have a look. And also, people have developed CRISPR-X, which is nothing but they have added MS2 hairpin with gRNA. So MS2 hairpin can hold many deaminase. So what happens? Wherever you target that, it goes there and can do many changes. So within a certain area, you can do targeted point mutation. Instead of one, you can do many by CRISPR-X. Also, people have used CRISPR. It's a new technology called Retron. So people use Retron. What is Retron? Retrons are natural DNA elements coding for a reverse transcriptase as well as a template on which the RT acts to create a multi-copy single-stranded DNA. So how this was used? So Retron is fused with gRNA. So now you have a Cas9 and gRNA, and then you attach a Retron, which is basically a template and a RT enzyme. Now the template, you can design that template, you can introduce your gene of interest there, and then this whole thing can go inside of the cell. Template can be made in front of the cut site, and homologous recombination can happen, and that template can get inside of the cell by this CRISPR technology, a modified CRISPR technology. Similarly, people have used Evolve-R. People have developed Evolve-R. What is that? So Evolve-R consists of an error-prone polymerase. So this one error-prone polymerase, you fuse with a nickase, nicking Cas9, which is nothing but cuts only one strand instead of 2 strands. So once the Cas9 cuts the DNA, it gets dissociated. Now error-prone Paul basically attaches there and try to rectify, and it gives rise certain kind of mutations. Now you can use many gRNA and target this whole system inside of the cell in many places of the genome. So your Cas9 will go and cut, Paul will rectify and bring different mutations. So you can generate a pool of mutants within very short period of time for your research. So this is called Evolve-R. So you can change in a tunable window, you can change randomly by point mutation, creating many point mutations. Similarly, people are using for gene knock-in. In case of plant, we know we use agrobacterium for plant transformation. And that agrobacterium has many Vir genes. So now certain Vir genes can attach to the right border. Now you take a gene of interest, attach a right border sequence with it. Then with a Cas9, you attach a Vir gene. So that Vir gene will bind with this sequence supplied by you. And then gRNA will take that whole thing in a certain area of genome. And in the cut site, you will get that gene of interest in larger copy. And that can be introduced inside of the cell by homologous recombination. So Vir-D2 mediated homologous recombination can also be done. Recently, people have found that certain kind of transposes, jumping gene elements can also do gene insertion. So this is Feng Zhan's lab from MIT. They found that certain kind of cyanobacteria, they are having this kind of CRISPR-associated transposes, which basically helps in gene integration. So a lot of happenings are going on, lot of additions, new development. And the last one, which I would like to mention, is prime editing. This is a new technology came out recently. People used in animal and human cell line, and now this has been used in plant as well. It's a great technology because instead of gRNA, you take a PEG RNA. It is a different kind of targeting. You take a PEG RNA and your Cas9 protein, attach it and with a reverse transcriptase. Now PEG RNA will supply your gene of interest, and reverse transcriptase will copy that. And by CRISPR-Cas9 activity, that PEG RNA containing DNA will be copied inside of your target area by reverse transcriptase. So this is a great tool people are using. So you don't need to do any break. You don't need to do any double stranded break. So off target or other kind of things are not there. It hybridizes in 3 places, not only gRNA, it hybridizes in 3 places. So off target is not that prone, not off target prone. No need of repair template co-delivery. So you are sending everything together, and precise point mutation and small indels can be done by prime editing. So it's most advanced modified version of genome editing, which can be applied for gene insertion greatly. And in plant, it can be used for many purposes, like small segment insertion, small segment deletion, introduction of multiple point mutation, introduction of replacement of desired amino acid, correction of undesired mutation, introducing premature stop codon, introducing single point mutation, etc. So with this, this is my last slide. A new happening are coming out. So artificial intelligence people are using for CRISPR application. Now, as I mentioned, you take your gRNA, you take your Cas9 protein, you send inside of the cell, it will cut and do the change. Now, how this change will come up? Because after cutting, cell will try to repair, there will be mutations. Now, there are ways. Previously, people thought that mutations are random. But nowadays, scientists have found that these mutations are not random. Cell also has an algorithm to basically change these or bring these mutations. So in Delphi or Sprout or Forecast, in silico tool software has been developed by different universities and institutes, where you can basically predict this change, and based on the prediction, you can manipulate your experiment. So there's great AI tools are coming up every day. Recently, there is a one-seq tool which will come up very soon from University of Harvard, which I have seen in one of the recent symposiums, where very precisely you can also predict if there is any off-target, which is very high throughput system. So this is where I finish my first part, where I introduced basic CRISPR, its applications, its recent modification discoveries for applying in animal and human. So 45 minutes I have taken already. So I may need another 15 to 20 minutes to finish the second part. Might be a little more, but if time is not there, I can squeeze a little bit. So now the thing is, in agriculture, how this CRISPR system is used and what are the greatness of this system, why it is used. Previously, what happened? You send a tDNA with your gene of interest, and tDNA stays inside. So your foreign element stays inside to make a GMO. But for CRISPR-Cas9, you send a tDNA get inserted in the genome. Then the tDNA will give rise to a CRISPR-Cas9 tool. The CRISPR-Cas9 tool will do the change, this red color area. And then in the next generation, the tDNA will be segregated out. So you will discard that progeny. You will keep that progeny which has only change. So what is there? What we have got here, no foreign element is there. No insertion element is there. You just make change in certain bases inside of the genome, and you accept that clean element or clean progeny or clean plant for your scientific purposes or application purposes. So this is the basic change CRISPR has bring, no foreign element, only your changes. So why this is so important? It has another one applications, a great applications. So if we do breeding in a traditional way, then we take our germplasm of interest, then we take important donor variety, we cross them, we get a progeny, then we do back crossing of that progeny for 5 to 7 generations, and then it takes 8 to 10 years to develop a new product or a new crop which can be used for certain purpose. So there are mutational breeding process as well nowadays. Worldwide, people are using it for many decades, and by using chemical or radiation, that also take 8 to 10 years. Transgenic breeding, if you want to develop GMO, that also quite time taking because the regulatory aspect is pretty long and it takes 8 to 12 years to make GMOs and bring in market. And you know there are a lot of issues with social acceptance, etc. But for genome editing, without insertion any foreign element or without keeping any foreign element inside of the genome, you can do it very fast within a year or two. And even if you use downstream integration breeding, you can do total thing within 4 to 6 years. And we see that regulation is much, much less hurdle for genome edited product in different part of the world, especially in US, in Canada, in Latin American countries, in Japan, and many other countries. In Australia, people are considering, the regulators and scientific boards and bodies are considering this simple genome editing, which resembles natural mutation as natural product. So you don't need to go for lengthy regulatory process and cost. So there are mainly 3 types of editing can be done in agriculture by this genome editing. First is SDN1, where you send the genome editing tool, you cut, cell repairs, so you get a mismatch of small indel. The breeding equivalent, the native breeding equivalent, if we see, it is just by a mutational breeding. It's just type of mutational breeding. In rice, many trait has been developed by mutation breeding, like RC, FRG1, WAXC, XA13, male sterility, etc. So, which has been done by mutational breeding. You can do it within in 1 year by CRISPR. So this is for agriculture. The second type, SDN2 type, where you insert an ortholog via homologous recombination. So this blue part is your gene area which you want to change due to certain reason. So you give a 2 cut at 2 end of that region, and then you send your gene of interest, either developed from either taken from a similar plant genome, or you develop it, whatever it may be, or take from some other organism, and you insert it by homologous recombination, which is known as allele replacement or allele swap. And this is basically a similar equivalent of breeding technology called MABC, which is marker-assisted backcross breeding, backcross hybridization. We develop near isogenic line by this process, this breeding process, which takes, again, 8 to 10 years to develop. That can be done in 1 generation, in 1 year, and then downstream applications. So SUB1, PUP1, Saltol, AG1, XA genes, PI genes, these are very important characters people have brought inside of the rice system by using this marker-assisted backcross breeding, by working 8 to 10 years for each of these gene applications, but which can be done by CRISPR in 1 shot. Now, the third type of genome editing which can be done is SDN3 type, which is basically, there is no breeding equivalent of it. It is just similar to GNA development, GMO development. But you can take a whole gene, you insert it by CRISPR-Cas9, and you get a GMO type of element, which is similar to GMO, but little bit more cleaner. You don't need to have left border, right border, all these things, less foreign elements, just your gene of interest by homology-directed repair. So Roundup or BT resistance kind of characters, which people have or industries has developed as a GMO, can be developed by SDN3 as well. So 3 types of genome editing can be used in plant: SDN1, SDN2, SDN3. So now, if you see this equation, this is basically breeder's equation for genetic gain. Breeder's equation for genetic gain. So whatever way you do breeding, or whatever way you do genome editing, or whatever back crossing, or you use this formula of genetic gain. Now forget everything, just think about breeding cycle and cost, L and C. So if you can reduce breeding cycle by twice, your genetic gain increases by twice. So CRISPR can do that. CRISPR can bring down your breeding cycle greatly, bring down greatly your breeding cycle, and that will increase your genetic gain. Also, it can bring down your cost significantly. CRISPR can also influence your genetic standard, selection accuracy, selection intensity, etc. I'm not going in that detail. I would concentrate here that for commercial breeding purpose, end of the day, you have to make seed, you have to bring that in market. Either you do it by public sector, university, institutes, government, or you do by industry. Whatever you do, end of the day, time and money matters. So CRISPR-Cas9 can greatly do good in that area with respect to our present breeding activities. So there is limitation. What is that limitation? So CRISPR in this area, this subtle area, CRISPR-Cas9 can work greatly. But at the bottom area, CRISPR-Cas9 still cannot be used. Why? So the complex traits, poorly characterized trait, CRISPR-Cas9 cannot be used. None of the system cannot be used. Trial and error breeding integration can be done and checked, but you need to understand the biology greatly. You need to understand the pathways greatly, and you need to understand gene-to-gene relations greatly to find out which gene to change and which gene to modify. This is less of a problem for genes like height determining gene, well-characterized disease gene, biochemical grain quality gene, physical grain quality gene, submergence, salinity, etc. These are all either single gene or few gene pathway which are well known. We can apply CRISPR and immediately change it. But for complex traits like yield, like drought, etc., where cascade of genes are involved, it is still a challenge. So we need to know biology greatly to apply CRISPR-Cas9 properly in agriculture. Already there are many CRISPR-Cas9 products in pipeline. Some got nod from the regulators, especially in US. This is basically a non-browning mushroom. So if you cut a mushroom or if you cut an apple and if you keep it on your table, after a few hours, it will get black. So only one gene product does that, which is called polyphenol oxidase. So by CRISPR-Cas9, one scientist in University of Pennsylvania knocked down that browning gene, and you can get a non-browning mushroom. So regulatory, etc., has been done now. This will hit the market very soon, and this has been considered in US as a traditionally bred product, similar to traditionally bred product. Similarly, beer industry, those who are using hops for hop flavor of beer, that hop gene in yeast you can modify by CRISPR-Cas9, and you can make hop-free beer. Hop requires a lot of water and a lot of processing to make beer. So you can get rid of that hop production and downstream application of hop. You can save a lot of money, a lot of time, a lot of water by just switching two couple of genes by CRISPR-Cas9. Similarly, a self-pruning gene has been used for large-scale food production in tomato, gluten-free wheat, waxy corn, decaffeinated coffee, spicy tomatoes, etc. People are using by changing single gene and giving rise a new product, which is really beneficial for human consumption or for market. So companies and industries and institutes are doing this, and in most of the countries, this has been considered as non-GMOs, and this has been considered similar to traditionally bred product and is used. Now, couple of general slides, how this CRISPR-Cas9 fits in the big picture of designer product development or commercially viable product development for government organizations or private organization. So whatever you do, you have to do breeding, right? So there are two things, molecular breeding and trait design. Both you can do. Then multi-generation genomic selection. And for trait design, you go from phenotype to genotype. You use omics and you use native traits and you make a product. Or you do the trait design from gene to phenotype through GMO pathway. We know issues with GMO. I'm not going into that. So in these breeding design, native breeding design and molecular breeding design of product development pathway, CRISPR-Cas9 can play a very significant role to bring a breeding product. So rapid functional validation of the target genes or target QTLs can be done by CRISPR. So once you know that, or once you know all the detail, then you can do diverse allele creation. Then diverse allele creation could be go to the rapid trait integration, and within a very short period of time, you can produce a product. Now, what are the requirements in this pathway? So rapid functional validation, for that, what you need a IP right or a FTO, freedom to operate, using CRISPR-Cas9. You have to know, or you have to learn or do editing, multiplexing, and homology-directed repair. And then you have to apply genomics to do this rapid functional validation. Then after that, in the next step, what you need to do rapid knockout or knockdown. That means modulation, gene from genomics, phenomics, bioinformatics. And then in the next step, what you need, regulatory modulation. You need to do allele swap, expression modulation, base editing. Similar thing you can verify by TILing or SDN3, but for genome editing, you do by genome editing, you know that, switch to TILing if you need. Then in the next step, you do allele swap and elite editing. Your elite line transformation is needed, molecular analysis is needed, and haploid induction is needed, and finally stack development is needed. In the rapid trait integration, you will need haploid induction and transgene-free integration, and you go to the commercial product development. So these are the stepwise requirements for applying CRISPR-Cas9, which we can visualize for commercial product development or designer product development for agriculture. So, and finally, the last two slides in this area, I would, we know that haploid induction plays an important role in commercial product development. Otherwise, you need multiple generations to fix a certain gene in both the allele, and it requires many years. So what commercial industries are using? They use a tool called haploid induction. So after you make your product, within one generation, you can make a double haploid so that all the genes in both allele are fixed. So how this could be done? So SENH3 or patatin-like one, DAD-like one gene, these kind of genes are there, which basically makes change in centromeric proteins. So if you knock down that particular gene, then what happens after fertilization? One chromosome, the male chromosome, falls off for SENH3. So you get a single chromosome progeny, which is known as haploid. Now you duplicate that. There are many ways of duplicating. So you get a double haploid. So you make a single progeny chromosome and you duplicate it. So you get a fixed line soon. There is a new discovery which Kellier and Kellier et al. Came up with. It's a great thing to change different bottlenecks and to introduce haploid induction and CRISPR together. And it's a path-breaking discovery, I would say. And it can be used in many crops where transformation is not needed. Why transformation is a big problem for breeding? Because elite line transformation is still challenging. Many crops are recalcitrant to agro or bialistic. So you cannot transform certain crops. What is the elite line? Even if you consider rice or wheat or whatever, you take certain like IR64, Nippon Bear, this kind of rice you can transform. But there are many elite rice lines, indica lines, still you cannot transform. So elite line transformation is a big trouble. Now delivery of this CRISPR product inside of your cell line is challenging, and traditional integration is lengthy. So these are the bottlenecks. This can be really changed by CRISPR-Cas9. How it could be done? You take a CRISPR-Cas9 made double haploid, CRISPR-Cas9 made haploid inducer plant. What happens? You take a pollen, which is haploid inducer pollen, and it has a CRISPR-Cas9 tool in it, and use that pollen to fertilize elite egg. Then what happens? This pollen is changed or mutated in a SNH3 gene. So after fertilization, and this pollen also containing CRISPR-Cas9 tool, after fertilization, editing will happen. So you will get an edit in the zygote. Then the male part of the male chromosome will fall off due to SNH3 mutation, centromeric region mutation. So at the end, what you will get? You will get a single chromosome haploid progeny. That haploid progeny can be doubled by making a double haploid in one shot. So where it is, what you are doing here, in one shot, in one experiment, you are making a haploid along with editing. So again, I am coming back. You have a pollen which has a mutation in SNH3 gene and which is also containing a CRISPR-Cas9. Take that pollen and go to your field and fertilize your elite line, which you cannot transform. Just you put the pollen inside of the elite line, and the next progeny which you get in one shot, it will be haploid and edited. Because the pollen has SNH3 gene modified, that due to this modification, the male chromosome will fall off in the zygote, only female chromosome will stay, and also that edited sperm pollen contains CRISPR-Cas9 that will do edit. So at the end, you will get a haploid line with edit, and the male chromosome will fall off, and it will carry along with, it will also carry the CRISPR-Cas9 tool. So CRISPR-Cas9 tool is removed, male chromosome is off. You are getting a haploid. You just double it, get a double haploid. So this solves a lot of problems. What it solves? No need of transformation. Just once you have to make this CRISPR-mediated elite line haploid inducer. Haploid inducer. That haploid inducer, and you collect that haploid inducer pollen. In this way, people have taken even corn pollen and pollinated wheat. You can get haploid wheat in one shot. So these pollen can be used in different purposes by elite line editing and elite line haploid production in one shot without doing any transformation or any laboratory-based experiments. So this kind of path-breaking CRISPR-based tools are coming up in agriculture as well, and people are greatly applying, industries are greatly applying these kind of things. I'm almost there. I need another 10 minutes. Do I have Shubhabrata?
Yes, you have. We have a 30 minutes up to your lecture, then the process. So up to 30 minutes you can use, no problem.
Okay. So what happens then worldwide? This is the regulatory status in present CRISPR crops globally. So Canada, Brazil, Colombia, and US, not regulated, because CRISPR-Cas9 mediated SDN1 is considered as just similar to native breeding products. So these, and also in Australia, in Japan, we all know. Then Europe, still it is banned, because Europe has given a very strange decision that any new mutagenesis technique comes under GMO. So that goes through lengthy regulation. Although they are not considering the chemical or radiation mutagenesis products as new mutagenesis product. So that is very strange. I know that many UK, Germany, France, these governments, UK, Germany mostly, and Spain, they asked a European Commission to reconsider this verdict. And in India, it is still under discussion. In China, it is still under discussion. Experimental activities are allowed, but product development is still under discussion, and both the countries haven't decided whether they will regulate it or not regulate it. So at least a big chunk of Latin America, US, Canada, and Australia, and Japan, you can use your genome edited product in the market as a non-regulated crop. And the last phase, the CRISPR water use efficiency, a case study I mentioned. So I completed the basic application of CRISPR-Cas9 in agriculture. And this is the last part, which we worked a few years back at International Rice Research Institute when I was a faculty there. And I worked with Professor Paul Quick, C4 Rice Consortium lead, and Professor Julie Gray from Sheffield University. We tried to apply, those were the early days of CRISPR in a plant. So in agriculture, we wanted to apply CRISPR-Cas9 and CRISPR-CPF1 to change water use efficiency. So we know this picture shows the stomata. We know in the plant, in the leaves, in the stems, there are stomates. Basically what it does, carbon dioxide goes inside through this. So photosynthesis is influenced by these stomates opening and closing. And also oxygen and water path for the plant are these same stomates. So stomata are small pore-like structure located on leaves, stems, surrounded by a pair of guard cells. And we know in a drought condition, what happens? You have very little water in the soil. So what plants wants to do? It takes the water inside of its body, but wants to keep it. It doesn't want to send that water out, because in water is scarce. So you don't have much water. You want to store as much as water possible inside of the plant body. So if you can reduce this stomatal number in plant, then less water loss will happen. More water will be stored inside of the plant body. If you see the crops or the plants which grow in the drought-prone area, they have less stomata. But the plants like rice, wheat, etc., they cannot grow in a drought-prone area or a very dry area. They have many stomates to do as much as water regulation possible. So we tried to change it. We tried to change it by CRISPR-Cas9. We used both CRISPR-Cas9 and also CRISPR-CPF1. That time CPF1 was new, not used in plant. So this is the stomata development pathway for dicot, example Arabidopsis or monocot rice. Different stomatal primordial cells during the developmental process of plant, different genes are involved in developing stomata, the guard cell, mature guard cell. So now we targeted this OS-EPF-L9. This is a positive regulator, also known as Florigen gene, a positive regulator for stomata development. So we targeted this gene. How we did that? We used CPF1 and Cas9 both. So in two different experiments, this is the gene of EPF-L9. So we targeted exon 1 by CPF1 and Cas9 both. We know that from the PAM site, Cas9 will cut in upstream, CPF1 cut in downstream. So we used Cas9 and CPF1 into different experiments, and we developed the edited plants. So we did sequencing, we did surveyor assays, and we found that there are edits, and that edits are getting transferred in the next generation as well. And then we did a southern blot analysis to see that the CRISPR-Cas9 tool is segregated out in T2 generation. So here you see this is the EPF-L9, CRISPR EPF-L9, where you can see this CRISPR tool is present, but in our edited plants, CRISPR tool is not present. So what we have an edited plant where CRISPR tool is segregated out, only some changes in the EPF-L9 gene. What was the effect of that? So on the top, you see there are yellow stars. These are basically stomates. this is a wild type. When we do CRISPR-Cas9 or CRISPR-CPF1 edit, we get edited plant, and there are only few stomatas. More than 8-fold decrease of stomatas we could develop in a plant, in rice plant, which by using CRISPR-Cas9 and CRISPR-CPF1. And when we use this plant for downstream analysis, we found that these plants, as there are much less stomata, they can hold more water inside. And as they hold more water inside, their water use efficiency is greatly enhanced. And that water use efficiency can be positively used for drought responsiveness. So similar activities, and also what we did, we tried to see if there is any off target or not. That time we used some available algorithms, some softwares. We targeted different first 20 possible off target regions. We amplified those regions, clone and sequence, and we found there was no off target in this particular experiment. So end of the day, what we received an edited plant. Only few base pairs are changed. So one gene is knocked down or knocked off. Due to that knocking off, the number of stomata got reduced to 8-fold. And this reduced stomata is the reason for more water storage or reason for positive drought responsiveness. So similar research later on carried out in Sheffield. They also manipulated, overexpressed and manipulated different other genes in that whole pathway. And they could see there are positive regulators, negative regulators. By knocking down positive regulators, you can increase water use efficiency by decreasing stomate numbers, and overexpressing negative regulator, again, you can decrease stomate number and increase the water use efficiency. So all this work you can see, our work you can see in this plant cell report. This was the early report where we show that CRISPR-Cas9 and CPF1 for targeting of a stomatal gene. And also you can see the new phytologist later on came out from Sheffield. We were also there, where they did a detailed understanding for drought tolerance by manipulating stomata using CRISPR-Cas9. Also that time, Virginia Tech University requested us to put a write-up on this in their website. So I wrote that particular time. So this is how we can, there are many ways, many pathways of improving water use efficiency. People are using it. It's very important for myriad purposes, different purposes of plant, not only drought responsiveness. It's a very important aspect, drought responsiveness, but there are many other things water use efficiency can manipulate for health of the plant. So this is one of the ways where by changing stomates, you can change water use efficiency greatly. So in conclusion, my last slide, what happens next? So I tried to cover discovery of CRISPR, basics of CRISPR, how that system works. Then I tried to cover how it is applied in animal, human, plant, industrial biotechnology. Then I tried to tell you guys about the agriculture and applications of CRISPR in agriculture. And then I showed a case study. But end of the day, it's a continuously moving field. A lot of new things are happening. But unless and until you use it for proper product development and bringing that product to the society, end of the day, it will be only scientific understanding, scientific knowledge. But we have to translate that knowledge so that we get more product in pharmaceutical, in gene therapy, in industrial biotechnology, in livestock breeding, in animal model development, and in agriculture. So if we try to summarize that, we will see that continuous improvement of the toolbox is needed. Mostly this is protein engineering, and people are doing it. Many new Cas9 and modified Cas9 modified systems are coming up. So, and gradually CRISPR is becoming an engineering platform, a great engineering platform. There are certain bottlenecks. This needs to be addressed, and scientists around the globe are doing it. Delivery limitations are there. I was mentioning about nanomaterial mediated delivery, different viral delivery. People are trying to improve those. Immune response, controlling immune response for animal and human health is important. Molecular precision is important. More and more editing efficiency improvements, so you don't need to do many experiments and reduce cost. And end of the day, large-scale manufacturing of all these products are very important. Next, delivery system engineering is also another one area people are working to improve, so that different delivery system, viral, injection-based, for agriculture, nanomaterial-based, or modified agro-based, whatever people are trying to do. So these are the areas continuous improvement is needed. And what would be the application pipeline in future, or where this field is going? So toolbox and protein engineering is the important part, as I mentioned. So different epigenome modification, for that protein engineering, or genome modification, for that attaching multiple protein molecules with the Cas9 and developing different kind of modified version. So this whole toolbox and protein engineering is very important. Followed by AI and data mining. Artificial intelligence plays a significant role here. So data mining, so huge biological data is available after successful genome sequencing. We all know that genome sequencing is very easy nowadays. Huge pool of data can be need to be curated and used properly. So artificial intelligence and data mining is extremely important. Prediction of off-targets, prediction of very unique gRNA, designing of very unique gRNA, prediction of edits done by the cell. So this kind of artificial intelligence-based tools are coming up. More and more will be needed, followed by manufacturing infrastructure. End of the day, this is how the whole thing will shape up. So you need supply chain, you need reactors for large-scale production of this CRISPR-edited cell, and identify and bringing out different chemicals or important important products for yourself. So automation, supply chain creation, and large-scale reactor system for fermentation and bringing out new products from CRISPR-modified organism is very important. So this is how I see the whole picture of the CRISPR-Cas9 field, its present status, and future perspective. Thank you.
Thank
so
you.
I for questions. So I took,
yes.
Oh, I took 1 hour, 20 minutes. Sorry.
No problem. We have a time. Should I minimize, end your slide, or you?
Yeah, I can end my slide, no problem.
Okay.
So
there is a chat box, and we pasted here few selected questions. There are a lot of questions. I will mail to you after the talk, but here we've selected few, and there is a lot of questions regarding the regulation in India, as you expected, and you clearly mentioned and by the graph that where it is and how the status worldwide. But still there are 2-3 questions on regarding this. And someone asks that whether CRISPR is a good topic for PhD. So it's also interesting. So I gathered few questions here. Can you see that in the chat box?
Yes. So first question, Dr. Kostap Panda wrote that what is the current scenario of production of single cell protein as well as nutraceuticals using CRISPR approach? Yes, lot of startups are there, mostly Boston-based and San Francisco-based, those who are working in these areas. And if you see some of the Netherlands company like VTT and others, they're greatly working on CRISPR-Cas9-based high-value chemical production, single-cell protein production. And I shown some of the, it would have been good if I could go back to my slide although, but can I do that? Okay.
You can do that.
Yes, so I'm not putting in a presentation mode anymore. Let me just get the, where are those? Can you see it? Shubhroto, you are mute.
Yes, I can see it.
Yeah, so, but I don't see the questions anymore. Anyway.
Yes,
so,
actually in presentation mode, this
yes,
box.
so I kept some of the examples in the early phase of my presentation where I showed applications in different fields. Yes, it's here. So people are using yeast, bacteria, algae, enzymes, plant cells, filamentous fungi through organism selection, organism development, process development, and scale up and piloting. And in the next slide, I kept this example of a VTT Industrial Biotech. This is a big company. They are working on different smaller cell lines, organisms to develop different nutraceutical chemicals. So this is one of the examples from Europe, but there are plenty startups and a lot of bigger companies. They have also started using CRISPR-Cas9 by for engineering and similar kind of activities. So yes, the answer is yes. There are many startups and many companies are doing, and we are also doing by using algal background. So from algae and cyanobacteria, we are also developing many new chemicals, novel chemicals, which we are using for CRISPR-Cas9 based organism-mediated, algae-mediated product development. The next question, Siddharth Mukherjee, hello, do you have some application notes in CRISPR in human personalized medicine with its clinical manifestations? Yes, so in my one of my slides, I enlisted CRISPR applications in cancer, CRISPR applications in different disease, genetic disease, infectious disease, then fertilization related, embryonic development related disease modifications, etc. So please check that slide. This will be available. All these slides will be available in the Bioingene website. So please check those. You have many examples and many informations there. Then Mr. Bipan, I don't know, Bipan, can we deliver CRISPR gene in useful fungi to control pest and disease? Yes, there are many papers where people are using CRISPR-Cas9 in fungi. There are many new, size is still a problem for algae and fungi. Cell sizes are very small. So if you use a big Cas9 protein, then it is difficult. But there are many smaller versions and modified versions of Cas9 people are using nowadays for introducing CRISPR-Cas9 in algae and fungi both. Mohammad Gulab Rubani, what he is asking? No, then where is that? Sorry. 3 questions. What is the editing efficiency rate of CRISPR-Cas9 in plant? Can you use CRISPR-Cas for protein engineering? The first question is, it depends. So there are papers from Yiping Chi's lab in University of Maryland, Shaixia Gao's lab in Chinese Academy of Science, IGDB. They show 100% CRISPR-Cas9 editing in crop like rice, wheat, a model plant like Arabidopsis, etc. Now, this depends greatly, which I believe, which from my personal experience, depending on the target you are choosing. So you know that genome and DNA remains in a very folded condition with different kind of structural formations. So if it's a apparently linear region, your gRNA can go and easily bind there. So in that cases, CRISPR-Cas9 works in a very high efficiency. But in a complex region of the genome or complex region of your, if the target falls in a complex region with different kind of folded orientations, then it becomes a little difficult and the efficiency goes down. Also, It depends on gRNA designing. You have to be very careful about gRNA designing, although nowadays different softwares are available. You can use those softwares for gRNA designing, and you get options of best gRNA and different other gRNAs. So depending on the specificity of gRNA, efficiency also varies. Pooja asked if CRISPR-based editing accepted in India, especially for agriculture purposes. Yes, I mentioned that in India and China, it is under discussion. A lot of deliberations are happening to the government from industry as well as from organizations like ICAR, DBT, DST, etc. Scientists came up with different kind of documents. It's under discussion. It's still not any decision has been made by the government how they will allow, even if they will allow it, either they will allow it as a native like US or Australia and other countries have made, or they will regulate as GMO. So that decision is still not made. Which class of algae are basically employed for biofuel production through CRISPR-based approach? Actually, all kind of algae. Mostly people are using this Chlamydomonas reinhardti for different kind of experiments. But for applying, making your industrial strain, different others are used based on your requirement. Some algal strains are good for novel biochemical production. Some are good for oil production. So it really varies. Dr. Jainul Abid Khan, targeting plant viruses using CRISPR-Cas either by genome editing or piecewise base editing, which one is more efficient for the generation of virus-resistant plant? Again, so virus resistance greatly depends on many techniques. So it depends on the virus, its genomic targets, which part you want to target. It depends on that. Nowadays, people are doing one very intelligent trick where they are basically taking different microRNA genes, changing that microRNA gene, and when that microRNA is expressed, it's basically give rise a modified microRNA, which can bind and destroy by RISC mediated destroying pathway the viral target, which can bind with viral target and destroy it. So microRNA based pathway, VSI RNA based pathway, and different straightforward targeting and neutralizing viral elements are done. So depending on the virus, group of virus, depending on the target region you are targeting, you can use base editing, or you can use large-scale CRISPR-Cas9, or different other modified, as I mentioned, modification of microRNAs people are using. So one patent is available online, which is called GEIGS. jigs. Please go through that. It's a new technology. One of the companies in UK, they are using it, and they have this patent, and in a selective way, they can change these microRNAs and basically target viruses. Next question, Pooja. Is it a good idea to work on CRISPR and PhD level? Look, CRISPR is a tool. Now, there are different applications of CRISPR. You can work for salinity tolerance in plant. You can work for disease resistance in plant. There may be many ways of identifying that particular problem and solve that problem. CRISPR is one of that. Yes, in today's world, it is the most modern technology. So definitely working with it would be a good idea. And second thing, there are also a field for CRISPR modification and CRISPR development. Labs like Jennifer Doudna, labs like Feng Zhang, or other associated like David Liu, Yiping Chi, all these people are working, Stanley Chi, all these people are working in the area of development of CRISPR tool. So that is also one area and could be a great PhD topic. Not one, many PhD topics actually. So the answer is yes. Sanjeev what is the cost for editing one gene by CRISPR system? Okay, exactly, I cannot tell you that, but it's very cheap because you just need a gRNA. It's just like designing a primer. So cost is very minimal, and Cas9 gene is Cas9 protein is commercially available. So that is also not very costly. So even I would say approximately with 7,000 to 10,000 Indian rupees, you can very much have one set of experiments and change one gene. So that is considered very cheap with respect to other systems. Kaushal Kumar Vati, what is your take on the regulatory aspect of genome edit crops in India? Is there any future hope for the plant biotech industry when National General Reliance are taking big lead now? What is your take on regulatory aspect of genome edit in India? Is there any future hope for plant biotech industry when? Yeah, I already answered this one because as I mentioned, it is under discussion, but definitely many countries has accepting it. And that's a great hope, unlike GMO, because GMO is still a great challenge and regulated in every country, even in US, where they are very positive about technologies. But genome editing in US, Latin America, then Japan, Australia, these countries, they have given green signal. So there is every reason to hope for Indian market as well. And I know China is, I was in China last 2 years and very closely worked with their regulatory divisions, government regulatory people I also worked with. So I know that China is investing highest in the world in genome editing for human health, genome editing for agriculture, but still they haven't. They are making a lot of products, but it shows that they will also come up green. It looks like otherwise the government would have not invested so much in this area. In India also, we feel the situation will not be like GMO. It will be much better than GMO situation. So I'm hopeful. The next question is from, is there any more question? Okay.
No, it's there is a lot of,
okay.
but I think it's okay. It's enough because.
Okay, so please send
yes.
me. I will be happy.
Yes,
yeah.
yes. I collect it. I collect all the questions. I will arrange and send you. Now I request Shoma, please do the interview section.
Hello, sir. It was wonderful to listen to you and so nicely presented. Thank you for coming here today.
Welcome thank you for inviting me.
Sir, we are now moving ahead to the interview section of today's webinar. And today is our 18th webinar of the series. And we are very honored to have you with us today. Sir, many of your viewers are watching from all over the world and in different places, in cities, in rural places. So they would love to know more about you. And you have been in so many places all over the world. And it will be very, enlightening for us to hear how you build your career. And could you please tell us about your journey, how you reached this position?
Yeah, so I had interest in biology from my school days. So I liked that. Although I wanted to be a doctor, but I was not that much attentive student during my 11 or 12. And later on, I switched to general biology. So while learning basic animal science, I got interest in molecular biology and related field. And that time, genetics was the early days of molecular applications. So later on, after some struggle, I realized the area which attracts me most is gene therapy or gene transfer. So either any kind of cell you take, plant or animal cell, how genes are inserted, how genes are transferred, that basically attracted me. And then I went to International Rice Research Institute for my PhD and worked on rice, gene transfer in rice. So After PhD, I went to US in one of the labs where I worked on basic DNA replication in plasmids, and then switched to University of California, Berkeley, and later on University of Kentucky, and worked on plant molecular biology, mostly targeting towards gene integration and gene insertion in plant, and also related plant viruses and how we could identify different genes in viruses, how we could sequence a new virus. This was my area of research. So I also work in the area of rhabdovirus, plant rhabdoviruses, and related molecular biology. So I continued my journey with the field which I really like to pursue, which is basically gene targeting. So I'm lucky enough to continue there. Even if I got good positions, postdoc positions in different fields, maybe renowned labs, etc., but I decided to stick on to gene targeting. Then I came back to India for a short period of time in one of the private companies, and I was their group leader for gene targeting and transformation. And later on, I got faculty position in ERI, and I was a scientist there and group leader. And that time, the talent was the big thing in the market. So I started working with C4 Rice Consortium with Dan Voitas and team. They're very well known in talent. Now they are having a company called, I forgot the name. So they are doing, so I started with talent. Then basically CRISPR started. I grabbed that, I switched on to CRISPR like that. Then again, return back to industry, first in Syngenta and now in Reliance. So end of the day, I am pursuing. So it's very important to understand what you exactly like. If you tell I like biology, it's very vast field. It has animal, human, plant, etc. Whether you want to solve one particular problem, a disease or something, that could be one area, or you want to work in the basic molecular biology and know the activities of DNA, RNA, protein, etc. Or you will do protein engineering, or you will target a gene targeting, or whatever the area you would like to do. So it's very important to find that out and stick to that. So that's really happened in my career. Definitely there will be a lot of ups and downs. I also had, but sticking to what you like is very important.
So true, sir. Wonderful to hear that from you. Sir, I have a question on food security. How important a role will genome editing play in feeding the billions in the future?
We all know about Green Revolution, 1960s, the Green Revolution, and that basically changed the global food scenario. Otherwise, by today, we would have seen a lot of famine in different parts of the world. But this Green Revolution and gave us big stride in yield, overall yield. But later on, last one decade, we see that it has reached a plateau. And also the overall farmer fields are reducing due to urbanization. And also you would see that farming population, the new generation of farmers, they don't like to farm anymore. They're coming to the cities and they are doing different kind of other kind of jobs. So farming community is also reducing globally. So we need to produce more, and population is increased humongously. So we need to produce more in a very limited farmland and limited input. So new technologies are extremely important to help plant breeding, to develop more products, more food. So who can help? Like genomics, bioinformatics, different other technologies. CRISPR is also a big addition and a great addition in that. And I believe with the help of these new technologies, breeding would be enhanced. It will be, its speed will be increased, and we'll be able to produce more product, more food in future by using all these new technologies.
So you explained that so beautifully. Sir, what role will CRISPR play in renewable energy?
Yeah, so renewable energy, a lot of things like, again, I will come to the same point. Wherever there is a cell, you have a DNA or RNA. As long as you have DNA and RNA, CRISPR system can modify that. So alternative energy, also great effort is going on either by different crop mediated biodiesel production or algae mediated production or different other microorganism mediated alcohol production, etc. So all these are organisms and have DNA or RNA. So you have chance to modify those by this new tool CRISPR. And modifying genes and making it more productive, making it more beneficial, that option is always there. So I truly believe that CRISPR-Cas9 and genome editing technologies will really enhance the production of organisms, which will basically give rise more and better version of alternative fuels.
All right. Sir, according to you, which sectors of plant science research will prosper in the near future?
It's very difficult to tell because all sectors are important. It's a continuous knowledge gathering and continuous discovery is happening in everywhere. Whatever sector one likes, if we concentrate on that very seriously, even if you do taxonomy of plant, if you do it greatly, you do molecular taxonomy, you go on doing it throughout your life, that also has a good career, and that field is also growing. So yes, some changes are coming. Every field is going through different kind of evolution, but all fields are important. Don't consider that we only have to do molecular biology, etc. And for commercial crop industry is concerned, if you consider with respect to job, etc., whatever modern thing you do, you get jobs. But everywhere, if you open a job site, you will see breeders are having jobs everywhere in the world. So different field has different applications, and all these fields are useful for product development. Not any single field is important. So whatever you do, if you do greatly and seriously, that will be helpful, and that field will grow. So I gave the example of taxonomy. When I was a student, a lot of people, I also was not very interested in learning taxonomy. But now when I went to US, I realized that molecular taxonomy and other taxonomy work is going in such an extent, and so much funding is there, so much research is happening there. So which I used to consider very boring, I realized that is also a great field. So that's where I'm coming from. So
all right.
Answers your question?
Sir, one final question, and you can conclude our interview session by listening to you, listening from you, a few words of advice or suggestions or encouragement to the new research fellows out there.
Yeah, so you should have a dream and follow that. What you want to be in your career, in your life as a scientist or as a biologist or as a professor or as a teacher, whatever you want to be, just consistently do that. There will be ups and downs. I also have gone through many ups and downs in my career and life. But if you really keep interest in something, just sincerely follow that. Some or other day, you will be able to do it as you wish to do. So that's it.
Yeah, it was so lovely to have you, and the way you explain is amazing. I mean,
Thank
many
you.
will be your fan from now on when they see
I
your
hope,
presentations.
although the title was mostly agriculture, but I brought like 50% of my talk in non-agriculture thing. So hope
It was
people
very
is
holistic.
heard.
It was beautiful. It was okay. So let's go
Thank
back
you.
to
Thank
now.
you very much.
Thank you, Shoma.
Thank you for inviting, Shubho.
It's most welcome, and we are very fortunate that I know you and your interest in CRISPR, because nowadays I follow your comment or your post because it's most updated. You sir, because I don't know about the prime editing paper, and you just posted in LinkedIn, so I go through the prime editing and then okay.
Media is becoming really helpful if you want to use it properly. So that's really great.
Yes, so I know
Thank
that
you. Thank you.
Whenever got any update in CRISPR world, you one of the such person that you catch it. And
Thank you to know that's helping.
yes, and
Okay.
thanks very much for coming. I will send you all the informations regarding the today's talk. We already received 1113 feedbacks, because these are the participants, those are interested on the certificate, but viewers are more than that. So we collecting the.
So how many people were there?
It is very tough, but I guess it's about 2,500. But when I give you our live feed and everything, because it's broadcast very social media, and so many people have come for a whole talk or for a section of your talk. And this talk actually shows live up to 1 and 2 hours, because it's streaming, and then it will shows that it is streamed, it was live.
Okay.
So we got the views from this section, and it remain open for all. Anyone can see it. So I want to thanks from the Bioingene that it is a great pleasure for us, and I am sure there lot of positive feedback I get, that your slide is very good, very understandable, very well coverage, and this is one thing you covered other aspect also, because now we can consider that Bioingene has different type of people, though it is based on plant, but so many people are interested in animal and bacterial system. So this talk is more relevant to them also. So
Thank you.
once again,
Hope everybody enjoyed.
Yes.
Okay. Thank you again.
Okay.
Bye.
Thanks for coming. I request Neha. Thanks, and I. And happy birthday, Neha, to you. Happy birthday once again. So enjoy your day. It's very tough to getting a birthday girl on the board, but you shows the high level of professionalism. Thanks for coming.
I love genomics. Yeah, thank you guys. Bye-bye.
Thank you. Bye-bye.
Have a nice day.
Same to you. Enjoy your day.
Bye. Have a nice day. Bye.
Thanks, Soma, for the help in the interview sessions and all. And I don't think you downloaded the presentation. Okay. So I can start the presentation.
Shall I leave now?
Yes.
All right. Bye, everyone.
So here these are the upcoming talks. So our next talk on 8 October. It is okay. This is a password. I think everyone got a link and password, and link is working fine. So don't worry. You have a half an hour and 40 minutes more time to fill the feedback form. I hope you guys enjoyed this talk. Our next webinar on 8 October, and all the registration are open through our YouTube, through our website. We are also available on YouTube, Facebook, Twitter, and LinkedIn. So you can connect with us in different social media also. And someone asking me whether there is any process of updating you through email. Is there is an option in the feedback form that whether you willing to get the notification from Bioingene or not. If you choose the yes, then we conclude you in our group, notification group, and we'll time to time we'll send you the email. It's nearly 2 or 3 email in a month. So this is our next talk. Then 13 October, there is another talk from Niti ma'am from ICGB. There is another talk from Dr. Dhanjay from ERI. Its talk is on rice. There is another in November. These are the talks in November from JNU Professor Ashwini sir. This is another talks in 24 November from Oliver. He is a cluster leader in ERI, Philippines. And as I say that we are available in LinkedIn, Facebook, Bioingene, Twitter, YouTube. Our official website is bioingene.com, but it is not yet developed. So we use our alternative website. But if you need some information, you can start from bioingene.com also, or you can just Google Bioingene. And we keep posting our all information in all the different platforms. And I recommend, or I request all of you, if you know someone is interested in plant or interested in such activities, please forward the information so we can got many actual required or many viewers on the field of plant biology or plant science research, which is our plan to promote worldwide. Today we got 63 countries registration from the 363 countries. It's day by day, it's increasing, and it's also very interesting and encouraging for Bioingene also that we are being recognized by many countries. And time, our time is Asian or content, or it is Indian Standard Time. Our talk is suitable for Indian Standard Time, but very slowly we try to cover that population from the different continent also. So being with us, this talk is available, but we can't give you the slide like PPT and PDF file, but this whole talk is available from now. So there is no problem to come and watch this talk again and again. And we have all the talks, all the interviews uploaded in our YouTube channel. Kindly subscribe this channel, or you can follow us any of these platforms. So this is the officially end of our today's webinar. And as I say, all the webinar are all webinars are included, but I'm very sorry that there is another webinar I forget to include here. It is on the 2nd October. The next webinar is on 2nd October. So it's from the 10 AM on the soft scale. So just to give me a minute, I want to search it. So there is a small correction. Our next webinar on 2nd October. It's not 8. 8 is a second one, but before that we have a 2nd October webinar. This is the part 2 of the soft skill. Yes, this one the next webinar. It is on the 2nd October. It was a continuation of our soft skill, because as we mentioned previously on the 5th September that soft skill is also very important for academia, for us also. So we are very fortunate that we try to convince and able to convince Dr. Kankana Ghoshal for her second continuation talk in soft skill. So all webinars are open for registration. Please do registration from our website. We keep open, it will keep opening just 3 to 4 hours before of the webinar. So you have a plenty of time, but I suggest as soon as possible, please register. So thank you. Now I'm going to end this session. We got around 1200 plus feedback form. And it's very unfortunate. There's so many people also asking for feedback, but it's actually the end of webinar. Please try to come at on time. Our webinar will start more or less the sharp time. So this is last time I'm giving the feedback form here.